Welcome everyone to our GloBIAS seminar series. Today, Anna Klemm will present. I will introduce her in a second. And before, I will just give some very short introduction to what GloBIAS is. So GloBIAS is a registered nonprofit association for bioimage analysts. And we have typically these boards like an executive board, which was elected in June 2025. So this foundation of the society just happened. And we have a couple of work groups working on publications, training and education, communication, and HPC cloud computing for image analysis, which is such a hot topic nowadays. If you want to become member, please reach out to the website and try to get involved with the community. So basically what we are aiming at is we want to build a sustainable base and legal entity to promote knowledge sharing, foster networking and professional development so that you basically can come from maybe a biologist, from a computer scientist, physicist to become a bioimage analyst and then become professional in this domain and network internationally. We also want to advance tools and best practices in bioimage analysis, and we want to develop and share high quality educational resources so that we do not have to reinvent the wheel whenever it comes to training bioimage analysis to colleagues. And of course, there are always new challenges in bioimage analysis, like artificial intelligence these days, and so on. Therefore, we organize these regular meetings and online events, like the one here today. Great that you are all here and that we can basically come a community and stay up to date with things. I would like to thank the Broad Institute for providing help with the infrastructure that we can use this Zoom link here, the Zoom infrastructure. The seminar will be recorded, and please ask the questions afterwards. We will then put the questions also on image.sc. And without further blah blah, I would like to introduce our speaker today. Anna Klemm did a PhD in cellular biophysics in the University Erlangen-Nuremberg, and then afterwards moved for a postdoc to the Max Planck Institute of Cell Biology and Genetics in Dresden, where I met her the first time. Oh God, Anna, where's time going? That's one or the other year ago, I think approximately a decade. Then she moved to Munich, where she became image analyst, working in a core facility and also giving lectures, teaching hundreds of students how to analyze image data, how to do microscopy. And eventually she moved on to the SciLifeLab, where she is now heading the Bioimage Informatics facility in Uppsala. So without further blah blah, Anna, the stage is yours.
Yes, great, thanks a lot. And I also still remember how we met at MPI. You were doing a Fiji macro for me, I remember, for helping me with a Fiji macro. So I will share a typical sentence. I will share my screen and you can shout whether this is fine. Do you see the correct slide? Yes.
Looks good.
Good. Then, yes. So thanks again for inviting me to the GloBIAS webinar seminar series. And what I thought to present is a bit live behind the scenes from the Bioimage Informatics unit, we call ourselves by now. And yeah, so of course, I guess in this audience, I don't need to explain that it's a very exciting and fast-moving field that we are working in. And I will also stress a bit more that it's actually also a moving job description, job description that we are doing. And then, so yeah, as an introduction, of course, science became much more multidisciplinary over the years. And then now to do a single project, researchers typically combine a lot of different tools and technologies and very high-end technologies, like starting, of course, in the wet lab, but then maybe moving to a super resolution microscopy or spatial biology or 3D fancy light sheet microscopy and so on. And then they need to analyze the data and maybe also obviously do some statistical analysis on them. And then finally upload data, publish everything. And of course, for all these steps, it's good to have experts that help you, that support you. And of course, this became a job over the years, and people are there at universities and helping. And in Sweden, so we are part of SciLifeLab, which is a national institution. And so there are researchers at SciLifeLab, affiliated to research at SciLifeLab, but then it has really a heavy focus on infrastructure. And there are around like 500, 500 infrastructure researchers. And I think most of them are permanently employed. And then, yeah, so it covers basically, we are covering all over Sweden and then located maybe in hotspots in Sweden. And then if you go to the SciLifeLab website, you see then like this plethora of units that help researchers with their research in different techniques, and really spans, like, as you can see, like genomics, proteomics, imaging, but also drug discovery, yeah, structural biology, and then of course a lot of data handling, data bioinformatics, and then we are part of bioinformatics as Bioimage informatics. And in Sweden, it's nice, quite well funded. I think Swedish government realized quite early on that this is important to fund, or to fund well infrastructure scientists and the support that researchers are having. And then and then also it's still also, for example, in this year, I think, I don't know, I had the impression that in many countries governments were a bit more careful in how they are spending money on research. And I think in Sweden they still increased the funding to the infrastructure at SciLifeLab. So this was very nice. So what you see here are numbers in Swedish crowns. If you divide it roughly by 10, a bit less, then you have, or a bit more if you divide it, and then you get to the euro. And that's money for 2025, 24 here in this diagram. And then, yeah, we are one unit of SciLifeLab, and obviously we are then doing the bioimage informatics part. So we are really one entire group, but not a single microscope, and analyzing data, and for researchers all over Sweden. And then you can see that, yeah, we support and give trainings and develop tools, and we really try to use kind of like the state of the art, what is around in the field, and bring this back to the researchers that they can use it in their projects, and then help also to, of course, deploy the computation methods that are around. And then, so as a facility, we have a scientific director, which is Carolina Välby. So she has her own research group in Uppsala doing image analysis, bioimage analysis as research. So we are having also we're very well connected on this part also. And she's very involved in the facility. And okay, so I told you that, yeah, we get nicely funded. And then, of course, as an infrastructure unit, as a facility, then comes with certain terms and conditions of funding, and then also how we report this. And this is what I show you a bit in the next slides. And so this I just recopied from the terms and conditions for funding from SciLifeLab that we get as individual units. And then you can see it, can maybe have not yet, no, I shouldn't do this. So I leave it, I think. We are supposed to facilitate world-leading research in molecular life science, and enable research that otherwise would not be possible in Sweden. And then, as a national facility, it's for multiple research groups that we cater basically. And then they also point out, for example, the good infrastructure practice and open access, fair GDPR requirements. And so this is kind of a package of conditions for the funding. And then of course, then we report every year our numbers, and I think many of you who are from facilities will probably know this. So what we at SciLifeLab typically then report is the number of publications that came out with our help, so either with us as authors, co-authors, or in the acknowledgements. And then we count how many PIs we have supported in one way or the other. Can be very short interactions until like hundreds of hours of support in building a pipeline for them. And then I need to move my videos a bit. So you can see then how this looks like over the years, they plotted them for us. It's from SciLifeLab, and like the numbers that we give to them and make a report to also an international advisory board. And then, the different researchers come from different universities and so on. So this, I think, probably sounds quite familiar to most of you that come from facilities. And then I guess you also know that this is not everything. So typically in a facility, what you try to do is also really be involved in teaching and give courses, and then also produce material for teaching. So we have a YouTube channel where we upload then webinars or also like tutorials on how to use, for example, Sepofiler or QPath or other Elastic and so on. And then we also are involved in tool development. I have a few slides for that. And then, we try to be very involved in the community in general, pipeline and development. And just to continue here a bit, so, I mean, to give a few examples, then, I mean, for the videos, as I already mentioned, so we have, for example, a tutorial on QPath where I saw, okay, there are a lot of people coming again back with the same questions of how to do something on this multiplexed image data. So I recorded a video, and then we also gave a course, and then we upload the material on GitHub, on a GitHub repo. And this is a different example. So here last year we were at the I2K conference, very, very nice conference. And then we participated with a workshop on also multiplexed image analysis, and together with even another lab from SciLifeLab. So it was really fun. Good. So this is a bit maybe, I guess, what a typical facility work looks like. And I think what I wanted to focus a bit is how do we select actually where to get active? I mean, we are an entire group of people, which is nice, but then still, of course, our time is limited. And at some point, we need to select what we are doing. And then I think I thought I start with this. So this is Erich Fromm, who was a Jewish sociologist, psychologist, fleeing Nazi Germany around 1930, I think. And then, so he has a lot of concepts about like activities, that activities can in reality be actually a passivity. So if you kind of run all the time to kind of calls that you get without having the chance to choose, it's rather a passive process than an active process. And then in a similar notion, he differentiates quite a lot between being an actor of what you're doing or just a reactor and reacting to others' need. And I think it's a bit maybe my main message that in a facility, maybe it's that you have so many user requests, and then of course it's very easy to get a bit in this reaction mode because you want to help. And I think it's a bit a call to try to steer your activities or to steer the activities in a way. And I took the liberty to cite Roberts and Johannes Softwedel's paper here. So you showed already some time ago that also a bit like this balance that you have to find in a facility in the different, like how can you support users? Like there are different ways, like for example, teaching or tailor-made pipelines or maybe a bit more general pipeline. And all of these have different pros and cons, and how to balance these. And you had already some thoughts on this here. And the way we thought of doing this is, of course, I mean, taking advantage of our staff, let's say, first of this, because I mean, typically in a facility, you have very experienced people and very engaged people also. And then I think through this job, we have quite good overview of what's happening and what is needed and what are the trends and so on. Like as an example now in the BIIF, I counted, and we have 6 members at the moment, and these are 46 years of post-PhD experience. So that's really quite a bit. And then additionally, as I mentioned, we are connected to Carolina Werby's lab, who of course has a lot of experience in more the research side of image analysis. And yeah, typically people, I think, in the image analysis field are very engaged, enthusiastic. And then the way we thought of doing this is I organized a retreat in the beginning of the year to basically bring these 46 years of experience in a room and really reflect on, I mean, first the activities of last year to see, okay, what projects did we have, but then also user requests that we already kind of see coming but haven't arrived yet. And then that would be very Sweden based, but then also like trends and like what is happening in the bioimage analysis field in general. Maybe even out, I mean, maybe not at the moment yet in Sweden, but will come. And then, I mean, in this retreat in February, we also worked on how to make how to reflect this in the workflows, like how to, when we take on projects. And yeah, since it's Sweden, we also had a sauna and a jump in a mostly frozen lake afterwards. So what came out of this retreat was, of course, a bit of clarity of where we want to get involved and what we want to do. And then we also updated our like standard operating procedure of how we take on projects. And this was actually also a bit in reflection on our Eurobioimaging guests, which I think I saw on the participants here, where we also discussed on how to handle projects well. So the idea is basically that to kind of steer, to have a bit more focus on when we get projects, like how can we really help to spend slightly more time and more brains. So we are typically 2, 3 people then in an initial meeting to know what to do with a new project, and maybe we can consult them to use a different tool, or maybe we can actually they would need to use a facility to improve their microscopy data. So we try to spend a bit more time in really selecting and planning how much time and which deliverables we want to give there. And then also, so this would be great when taking on the project, but then also in the course of the project that we really take time. We now have like a dedicated time slot where group members can present. I mean, I think we always have time, but it's good to have like really a time dedicated to this to present and get feedback by the others and take a bit of more time in thinking of how to proceed in a project in crucial points of the project, let's say. And yes, this is just to say that we try to have the projects in public GitHub repos, so following the FAIR principles also. Good. So this was, starting on how to try to select in a bit more conscious way how we work. And connected with this is also, are we, like, now that we are responsible for entire Sweden, like how do we make sure that we don't miss user bases, or who are our users, and how to steer this, who contacts us. And I mean, it's not, sometimes you cannot really know whether really reach all researchers that should be able to reach you. But in theory, we, as a national facility, of course, we are responsible for all researchers over Sweden. And then what happens in practice is that we help, and typically then people are happy, and then their neighbors are contacting us as well. So it's of course a recommendation, and then of course we try to be on conferences and participate also in PhD courses where you see more projects. But maybe that's still not enough. So I think What we then also try is to make it really visible what we are doing. So we have a project database, so people that have really no clue that there is something like a bioimage analysis support can get an idea of what this at all means. So they, like we ask every project owner, basically, we ask to give a small non-confidential picture and a description, so at least other researchers can get an idea of what can be achieved using image analysis. And then what I found also actually quite nice to screen for trends and needs is maybe sometimes a job that you sometimes avoid to do, which is like reviewing things. And like in my case, what I found, for example, useful is we are part of NBIS, which are the National Bioinformatics Structure in Sweden. And then their PhD students can apply for advisories. So also very nice program. So they get a staff scientist that helps them, I don't know, a few hours per month. And then, of course, there are more applicants than one can offer help to, but there you can see the trends. And the same is, of course, true when they apply for extra funding or then, yeah, in courses. Now there will be in a few weeks the EMBO practical course for advanced methods in bioimage analysis, and there we had like 190 or so applicants. And then, of course, it's actually quite nice to see what there are many PhD students, many staff scientists, and then they write a bit what they are doing, right? So like 3D organoids or whatever, and then you see a bit the trends, and especially if you do this over several years, you can see some trends. And then obviously you would go to conferences and join the GloBIAS seminar series and so on, but I think I don't need to tell you that. And then I think we try to also steer it a bit by having a newsletter, by then also, of course, presenting to make people that maybe are already aware of us, but we can make them aware of very specific pipelines that we've developed or very specific videos that we have produced. So we really try to have a bit of outreach there. Good. And then like another corner where we are, of course, active is really in active technology development. And again, it's, I think, different for different facilities of how much funding and time you can spend on this. In SciLifeLab, actually, we can spend around 20% of the funding that we get from SciLifeLab in a reasonable way, of course, for the tool development, technology development. But then we can also actually actively apply. So again, a way to actively steer what we are doing is to then apply to special calls. And this is, and of course, not only us are doing this, and it's really a shout out to the bio people at EPFL who do a really amazing job as an example, but I like a lot their QPath extensions, and they do I think see the needs, what is around, and then they make an extension that you can use in QPath. So I think I really like this involvement in the community needs. And then, yeah, from our side, I already mentioned the tissue maps project in the very beginning. So the tool where the facility is involved in co-developing is tissue maps, which is a GPU accelerated viewer for like spatial omics data, let's say like millions of data points that you can view with them. And this was started in Carolina Belby's lab and still is active there. And so it's a common project, Carolina. And this is like 2 slides of advertisement for tissue map. So you can see this is like an in situ sequencing data set with like more than around 2.5 million data points that you can inspect and zoom in, zoom out in a Google Maps style of fashion, and then export some regions of interest and so on. And then also it is primarily a viewer, but then it works via plugins where you can then add functionality. So for example, here you can like cluster then the genes by their kind of functional neighborhood into different regions by using a plugin called points to regions. And then, yeah, so this is then indeed one part, like coming back to our daily business. So this is now at the moment really an activity. So we are currently refactoring tissue maps since it's just a code base that has grown over years. It needs some cleaning and some renovation, let's say. And then it should be easier to facilitate community contributions. And then also, again, I wanted to stress that here also we really applied for extra funding to have functionalities enabling mass spec imaging, visualizing of mass spec imaging data, and then also connection to the spatial data format. So again, we kind of really try to steer a bit what we can do in our time. And then coming already a bit to the end, it's kind of what I think is important is, of course, the people and also I think the way you interact with other infrastructure specialists that are around you. And what we see more and more are these kind of dynamic teams that work on something, and then you form another dynamic team to form on a different project due to this multi-modal modality of the analysis. So what I mean with this is, like, as I said, we are part of SciLifeLab, and then you have really a lot of people there. We are part of NBIS, which are experts in bioinformatics, so for example, single cell sequencing, but also spatial analysis of in situ sequencing analysis, for example. And then we are also part of the Department of Information Technology, which has a lot of knowledge in image analysis. And then, I mean, often now we really form teams where it's like someone from us who does, like, let's say, the co-registration of images and consults in the parameter extraction. And then in the same really team, you have a bioinformatics specialist who then does the downstream analysis of this. And then these kind of teams can change over time. So I think that seems to be a bit where it will come more often, more kind of these bit flexible constellations of groups working on something. And then, yeah, so I included this slide for infrastructure staff because I think it's a bit of a shout out that it's a nice job to do, but also I think the staff is really, of course, the potential because I mean you have again you have these amazingly skilled people with a lot of enthusiasm, and I think and coming back again to this guy, the Erich Fromm, I think he has this book To Have or To Be also, and it's a bit of do you do things to I mean I don't know now in this context would be maybe to collect titles or something, or is it more to really work on something that you find interesting? And I think in the infrastructure world, you have really a lot of people that really drive on that they find the scripting cool and the extracting of information and visualization of the data and so on, so that they really like the work per se and not so much dependent on what is around. And this is, of course, the nice thing is that you find this then in many places. And I think GloBIAS is really an amazing place where you can meet really many of these people. And I mean, whenever I go to any conference course where there are many image analysts, I'm really every time touched of how nice people are and how inspiring it is to connect. So I'm really urging all of you to become a member in GloBIAS and become active and meet more of your colleagues and to connect. And I think, for example, when there was still Twitter the thing and people were tweeting from conferences and courses, then I found it always funny that or amazing that in bioimage courses or conferences, then this emoji would be used quite a lot. So it was the signature emoji of bioimage analysis was this exploding, amazing emoji. So I think that reflects very well how it is working in bioimage analysis. And yeah, so with this I come to again a slight advertisement, but I think it's with a good reason. So there will be SciLifeLab will push even more the staff scientists, and especially I told you that they hold these units doing amazing technologies, and they want to link even more how or to drive even more how they are linked and how the image analysis not images, not only image analysis, data analysis of these units is done. So they are hiring very soon. There will be 15 staff scientists hired, and so the information will be out soon. There will be a few which will have a focus on image analysis and then image data handling management, image data management, but also like more bioinformatics touch and so on. So please check it out if you're interested, and pass it on to the more bioinformatics friends that you have. And with this, I come to a summary. So I really think that, I mean, of course, this support, like research has become much more reliant on this support by high-skilled staff scientists, which is very good. And I think the importance of these people will be even more in the future, with more this disciplinarity of modern research. And as a facility staff, I think we can be with a bit of self-confidence. And then, I think we have both the responsibility, but also the power and the joy to steer the path a bit, like how we give the support and what we find reasonable and good in the way the support is given. And yeah, I'm open. I'm happy for any questions.
Thank you very much, Anna. Thanks for this great presentation. We actually have a handful of questions and we will start them immediately. But if you have any additional questions, the attendees, just write them in the QA box and then we will quickly go through them as long as we have time, and remaining questions may then be answered later on image.sc. So first question by Beth was, the project database is a cool idea. Is submitting your project mandatory or optional?
It's highly encouraged. So we kind of say it in the user agreement, but of course, and most people do it, but of course, if someone's really very much on, no, this is confidential. I mean, we still try to push them to say, yeah, you can write it very general, and then it's usually fine. But if they really don't want, then of course we don't push them. It's okay, but it doesn't happen too often. It's quite nice.
That's also a good advertisement, right, for the individual labs research.
And it's also, I have to say, it's very convenient because we also ask them directly for the non-confidential picture that we can then use. And then, I mean, for presentations like this or for outreach, it's of course super handy if you don't need to. I mean, we take then really the pictures they gave us and so on. And then it can be very handy. Mm.
Yes. Okay. Second question. For the other countries that are less developed in the area, at which level do you think a bioimage analysis facility should be? Should it eventually be developed into a BIIF, like a national format, or should one facility for each lab, department, institute would also work in the long term?
I mean, I think realistically it starts often locally. I think to me it makes a lot of sense if you connect, because many questions will be not only concern your own group, but will be, as soon as you try to do something a bit sustainable, then I think it will also address will also be interesting for other people, for other facility staff. But then, of course, realistically, you have all these, okay, how should it then be funded and who owns the person and so on. And unfortunately, this is sometimes hard to overcome. But I think maybe there's also there's, I think, a certain openness also to argue for like a good with a good reason, let's say, for good science, hopefully. I mean, I think if it's not that developed, of course, it's good to, I mean, even having at all bioimage analysts is still not so common in all places, so it's definitely good. And having already someone who knows where to point people to and which are the resources online is already goes, I mean, that goes already very far. So it's not, shouldn't, I mean, that's already amazing. So yeah.
I would actually have a follow-up question on this one. So a national core facility with a lot of staff positions, like permanent positions, is not the usual concept we find on our planet. I was wondering, depending on the degree of you were involved, but do you know how decision makers were convinced to do this on a national level with permanent positions? Was there a killer argument or something? Do you by chance know?
I mean, I think so. How it started, I'm not sure. And then, but what I know, what helps is so because I mean, legally, we are still employed by the local university. So I'm, for example, an employee of Uppsala University. And then, of course, they are, I mean, Sweden is very good with the permanent positions, but they are also quite strict. So you need to prove that you can fund a person for quite some while, that you have secured funding. And then, of course, if you have a huge body behind you saying, this is like the money that we get per year, then this, of course, helps to convince to
Mhm.
the permanent positions. And then how much, yeah, I think. Yeah, no, I cannot say so much of how it because of course, yeah, we are not involved in the teaching, master level teaching and so on. But I think we still bring overhead to the university, so they are maybe happy about that. And I
Mhm.
guess it's some balance.
Good. Thanks for your insights. There's a question also from Marco. Could you elaborate a little more on how you manage cross-unit projects, data infrastructure, task coordination, project management? Is every team coordinating themselves, or is there a unified platform strategy?
So we have, let's say now for the, I mean, in SciLifeLab, all these units have their own management. And also the data infrastructure is still a bit on, or still a lot on the under construction, let's say, a unified data infrastructure. But the, let's say, for example, within the bioinformatics, then when we have these big mixed projects, then I guess what we try to do is still to have a main manager. But then, of course, if it comes to really the specialized knowledge, then you have a bit of a local manager that then takes over this, and then you bring it back to the main, yeah, to the group again, and so on. So it's a bit flexible, and I think, yeah, the not so easy solvable task is how to, I mean, yeah, how to not have too many meetings is not so easy to solvable task because you need to, or you, because yeah, I mean, you want to meet, of course, and you want to know what people are doing. You want to be aware of what people are doing, but of course, you don't want to spend now joining the weekly meetings of 5 groups only because you have 5 modalities that you are combining. So. Mm.
That's indeed a challenge for many of us.
Mm.
I also like a lot the next question. How often does it happen that you have to refuse to help a user project? And what do you do in that case, and how do you explain it to the user? I like it.
We are still very nice, I think, but I mean, sometimes I think so. What we kind of agreed on is that we always try to help with a consultation, and then yeah, I guess still in many cases we are taking the project also on. But in what we definitely do is that we point them, as I said a bit, we point them to resources that are online, or when we really see the images are not that great, for example, or they used, I don't know, they used something in the microscopy which we don't agree with, then we raise this problem, and then we also really try to be that well connected to the image facility, like the microscopy facilities, that we can point them quite easily to the microscopy facility of the individual university, for example. So we kind of maybe pass it on sometimes. But I think, yeah, I think our aim is to really at least give some input and not leave them completely lost.
Good. Next question. So there are still something like 2 or 3 to go. I hope that's okay.
That's okay.
Alessandro asks, I may have missed it. What is the career progression like at SciLifeLab? What types of indicators, and then in brackets, technical or managerial, I'm not sure if this is a word, are used for promotion?
Yes, so this is indeed something they're working on. Also a bit, again, then you see a bit that it's different universities being combined to one big national institution. So I know that Karolinska in Stockholm, they now at least have different career, like staff scientists, 2 levels, like a staff scientist, a senior staff scientist. I'm not completely sure how they call it. And then I know that you get more salary, and the indicators, I think I'm not completely sure, to be honest. And I'm also not sure whether it was, I forgot a bit. So I mean, just now thinking what comes to my head, I think, I mean, still,
It's perfectly fine to postpone a question and say you will give a
yeah.
proper answer
No,
on
it's,
image
I
SEO
think
later.
publications probably still play a role, but it probably has more focus on then also acknowledged publications where you are acknowledged, I would guess. And then maybe if you have some tools, then you can probably also say the whatever downloads or something like that. But I'm not completely sure. But I know that there's some movement into at least have a slight career progression.
Mhm.
If you, I
Good.
mean, if the progression is not expert to manager, then that's it. Yeah. Sorry.
I also like the next question a lot. How is AI impacting the work at the bioimage analysis facility? Do you see the need for HPC resources that are available to run the pipelines apart from the tools?
Yes. Next question. Yeah, so it's yeah, it's AI is impacting our work by now mainly still by these pre-trained networks that you can or pre-trained models that you can use. Like I mean, Stardist, Cellpose, these things. SAM we also started using quite a lot. And then we did have a user recently that came and had already a Fiji macro and said wanted some input into a consultation round, and then we said, I think the story was Jonas from the facility. He said it's better to analyze slice by slice, and then he asked ChatGPT how to do this, and then it was not that bad the outcome. And then we again had a look, and then said, I don't know, we indeed had some input on how he was clipping his data by mistake and so on. So we still had a role in guaranteeing the quality. So I think this we do see. I would say I'm still quite amazed of how much we still use very classical image analysis and not so like the fanciest deep learning method. But on the other hand, it, yeah, what we also see is a bit more different modalities where then suddenly it becomes a bit more relevant. Like now we see more environmental pictures of, I don't know, pictures of beetles and something like that. And then suddenly we are using a lot of the segment anything for getting those and so on. So I think it changes also. And then HPC resources, yes, I mean, it also come so the HPC resources is indeed more and more needed, and then it's a bit old style still that then researchers will have to apply for access to a HPC resource, and we can help them to set things up, but then it's still a bit of a bureaucratic process, and what is needed is a much more easier access. And But then with these 15 new staff scientists, actually one task is also that indeed more tools can run on HPC resources on the data of the units of these SciLifeLab units.
Okay, cool. So I will skip one question and come to the last one, which comes from Asta, and she was basically asking, it's great to see that your facility nicely bridges biology, bioimage, informatics, data management, omics, even IT and other departments, etc. But what was essential for your unit? As in, is there a space for only biological image analysis facility? Any killer arguments here?
So if you have only image analysis and don't do the other things, you mean? Or whether they have a space? Yes. I mean, so I think there's, in general, it's moving somehow quite dynamically what we work on. And also what we see is that you suddenly have more local specialists suddenly that then do the spatial omics by themselves and so on. So this is very dynamic. And then, for example, at the moment, it seems like suddenly the 3D fluorescence images seem to be again more popular and so on. So there I would also see a only biological image analysis facility then be very happy to do these things. Was this the question? Am I
I guess
hitting?
so.
Yes.
I guess so.
No, but I
Gut.
think that I didn't really add, and I had it in mind that I see more more local analysts arising, and then also we can distribute our support also by helping them, giving a few consultations, a few hours of consultations to people that are already quite skilled, and then you can also scale quite nicely your efforts. And the same is also with nice tools that are around, right? More and more microscopes come with image analysis tools already inbuilt, and then also there you need to handle those.
Good. I would like to thank you again, Anna, for this nice talk, for answering all the questions. I will just quickly share this final slide as a quick advertisement. The next GloBIAS seminar will be in roughly 3.5 weeks by Jean-Yves Télevez from Institut Pasteur about image analysis services and scientifically added value of such a core facility. And with that, I would like to thank you everybody for attending. Again, if you want to become a member of GloBIAS, reach out. And last but not least, again, thanks for Anna for presenting and also Krista in the background for organizing. Have a great day, everyone.